Today, we are taking on a foundational mission. We're going right back to the beginning to master the absolute fundamentals of biology.
We're diving into the living world.
Exactly. We want to really unlock the sheer scale of biodiversity on our planet, and maybe more importantly, understand the scientific toolkit we had to invent just to begin studying it.
It's a fascinating problem, really. It's the ultimate exercise in trying to impose order on, beautiful chaos. The chapter text pulls you in right away with this sense of awe.
It does.
Whether you're looking at a galloping horse or the molecular traffic inside a single cell, that amazing diversity is the starting point for everything.
And that sense of wonder, that awe, it forces this huge question on you. What actually is life?
Right.
The text points out that this question kind of splits in two. There's the big philosophical one, what's the purpose of life? And we're definitely leaving that to the philosophers.
For today, at least.
But the second part, the technical scientific question, that's our focus. What characteristics truly define something as living as opposed to, say, non-living?
And you can't even start to answer that without grasping the sheer size of the problem. When we talk about biodiversity, we're talking about a known, described range of species somewhere between 1.7 and 1.8 million.
That number is just staggering. 1.8 million.
And that's just what we found and named so far. Think about it. 1.8 million different ways of being alive, all needing to be cataloged. Early humans, they saw this too, but their classifications were based on utility.
What can I eat? What can I use for shelter? That's
exactly, or they just deified it.
Yeah.
But that human-centric view only gets you so far. For real scientific progress, you need systematic description, and that's what drove the development of the whole system. Identification, nomenclature, and classification.
Okay, let's unpack that system. But I feel like we can't really start without talking about one of the giants mentioned in the text, someone who helped define the very unit we're trying to classify.
Oh, you have to talk about Ernst Mayr. Absolutely indispensable. He's called the Darwin of the 20th century.
And that is not a title you get by accident.
Not at all. He was this Harvard evolutionary biologist, and the text highlights that he almost single-handedly made the origin of species diversity the central question of evolutionary biology.
So before we can even name an organism, Mayr is forcing us to properly define what a species even is.
And that's his legacy right there. He pioneered the biological species concept that we, largely still use today. It's not just about looking similar, it's about a population that can actually interbreed and produce fertile offspring. He defined the unit of classification itself.
And he was certainly recognized for it. The Triple Crown of Biology, as they call it, the Balzan Prize, the International Prize for Biology, and the Crayford Prize. An incredible thinker. He died at 100 years old.
A full century of thinking about life. Now let's connect his idea of a species to the practical chaos of naming them. Why do we even need these strict rules?
It's the communication problem, right? I call it a mango here, but someone a few hundred miles away might call it something totally different.
Or worse, they use the same word for a different fruit. It creates inescapable confusion. Imagine trying to collaborate on research. You'd never know if you were talking about the same organism.
So standardization becomes a scientific necessity. The whole process is called nomenclature. We need one name for one organism, and it has to work everywhere on the planet.
But before you can give it a name, you have to do the crucial first step, identification. You have to be absolutely sure what the organism is. You describe it correctly, compare it to what's already known, and only then can you attach a name.
It's the quality control step. And to enforce this globally, you have these international bodies.
Right. The International Code for Botanical Nomenclature, or ICBN for plants.
And the International Code of Zoological Nomenclature, the ICZN, for animals.
And these codes guarantee two very simple but very important things. One, each organism has only one name. And two, that name has not been used for any other known organism. It's unique.
The architect of the system that makes this all possible was, of course, Carl Linnaeus. His system is binomial nomenclature.
Brilliantly simple. Every name has two parts. A generic name, which is the genus, and a specific epithet, which denotes the species.
Let's use the classic example from the book, the mango, Mangifera indica.
Perfect. Mangifera is the genus. Indica is the specific epithet.
But to use this system, you have to follow 4 universal rules. And these are not suggestions, they are. They're law in biology.
Rule 1 is about the language. The names are Latin or they're Latinized, no matter where they come from. That's why they're always printed in italics. It's a signal.
A signal that it's this universal, stable language. Rule 2.
Rule 2 is just the structure. First word is the genus, second word is the specific epithet. Simple as that.
Rule 3 is about how you write it. If you're handwriting it, say in your notes, you have to underline each word separately. If it's printed, you use italics.
Both methods just indicate the Latin origin. And finally, rule 4 is capitalization.
Right. The genus always starts with a capital letter. Mangifera. The specific epithet always starts with a small letter. Indica.
And you sometimes see that little abbreviation at the end, like Mangifera indica Linn.
What does that tell us?
The Linn stands for Linnaeus. It's an author citation. It just means he was the first person to describe that species according to these rules. It's a nod to the history of the discovery.
So we have the rules for naming, but even with names, studying 1.8 million species one by one is, well, it's impossible. So we have to group them.
And that's classification, the process of grouping things into convenient categories. And those categories are based on easily observable characters.
And the scientific term for these categories is taxa. Singular is taxa.
And what's key to understand is that taxa can exist at all different levels. Mammals is a taxon, but so is dogs. They're just categories at different ranks in the system.
And this whole process, characterization, identification, classification, and naming, that is what we call taxonomy.
Modern taxonomy is incredibly detailed. It looks at everything from external and internal structure to the development process and even ecological information. It's a huge step up from the early human classifications based just on, simple utility.
Okay, so if that's taxonomy, the process of grouping,
Yeah.
where does systematics fit in? Linnaeus titled his publication Systema Naturae.
That's a great question, and the distinction is critical. Taxonomy is about the what and the how naming and grouping based on similarities. Systematics takes it a step further.
How so?
Systematics includes all of that: identification, nomenclature, classification, but it adds one more crucial layer: the study of evolutionary relationships between organisms.
So it's not just asking what are you, it's asking who are your relatives.
Precisely. It puts everything in an evolutionary context, and that context is what builds the entire framework we use, the taxonomic hierarchy.
A hierarchy of steps where each step is a rank or a taxon.
And here is the single most important rule to remember about this whole hierarchy. As you go higher up, from species towards kingdom, the number of common characteristics goes on decreasing.
The lower the taxon, the more things the members share. The higher the taxon, the more general the similarities become.
Exactly. Let's walk through it, starting from the bottom.
Okay, the most fundamental level is species.
A species is a group of individual organisms with fundamental similarities. We can tell them apart from other closely related species based on distinct morphological differences. Sapiens for us, Leo for the lion.
Then we move up to genus.
A genus is just a group of related species. They have more in common with each other than they do with species from a different genus, like the potato and the brinjal. Different species, tuberosum and melongena, but they're both in the genus Solanum.
In the example with the big cats.
Right. The lion, Panthera leo, the leopard, Panthera pardus, and the tiger, Panthera tigris, all belong to the genus Panthera. They're very different, but they share features that group them together and separate them from, say, the genus Felis, which contains smaller cats.
Okay, above genus we have family. Now we're grouping related genera.
And the number of similarities drops again. In animals, the family Felidae includes the big cat genus, Panthera, and the small cat genus, Felis. But you can easily tell the difference between the cat family, Felidae, and the dog family, Canidae.
The differences become more obvious at this level. Next up is order.
An order is an assemblage of related families, and again, they share even fewer similar characters. A great example is the order Carnivora.
Which includes?
It includes families like Felidae, the cats, and Canidae, the dogs. They are quite different, but they're grouped together based on shared characteristics like being carnivorous mammals.
Above order, we get to class.
Class includes related orders. So, for
Yes.
example, the class Mammalia contains the order Carnivora, which we just mentioned, but also the order Primata.
Which is where we fit in monkeys, apes, humans.
Right. And what do we all share? Very broad characteristics, like having hair and mammary glands. The similarities are becoming much more fundamental.
Almost there. The next step up is phylum for animals or division for plants.
And this is a huge category. Phylum Chordata, for instance, is massive. It includes the class Mammalia, but also birds, reptiles, amphibians, and fishes.
So what on earth does a human share with a fish that puts us in the same phylum?
It boils down to really core structural features. For chordata, it's things like having a notochord at some stage of development and a dorsal hollow neural system. Very basic body plan stuff.
And at the very top of the hierarchy.
Is the kingdom, the highest category. All animals from all the different phyla are grouped into kingdom Animalia. All plants go into kingdom Plantae.
Let's use that table from the book, table 1.1, just to drive this home. Look at man, Homo sapiens. Genus is Homo, family is Hominidae, order is Primata, class is Mammalia, and phylum is Chordata. By the time you get to phylum, the only things we share with, say, a shark are that notochord and neural system. The similarities have diluted almost completely.
It's the perfect illustration of the rule. The higher you go, the fewer characteristics are held in common.
So after all this meticulous work, imposing this structure on 1.8 million organisms, what's the big takeaway? Why does this matter outside of a biology exam?
It matters immensely because taxonomy isn't just an academic exercise. As the text summary points out, these studies are essential for agriculture, for forestry, for industry, for all kinds of bioresource management.
You can't conserve what you can't identify and classify.
You can't. Knowing what we have and how it's all related is the first step to everything else in biological science. It gives us a universal language to talk about life on Earth.
So every single organism, from a bacterium to a blue whale, has a precise, agreed-upon place in this system, a taxon.
And it's a system built on rules for identification, naming, and classification that are accepted globally, but it's a system that's also constantly evolving.
And that brings us back to Ernst Mayr, his biological species concept, based on who can breed with whom. It works wonderfully for a horse or a mango tree.
Absolutely. Clear lines. They can breed or they can't.
But what about the organisms you can't see? Bacteria, for instance. They don't reproduce sexually in the way Mayr defined. So how do you define a bacterial species?
That's the challenge, isn't it? For them, the concept has to be based more on genetic and metabolic differences. The rules have to adapt.
So here's a final thought for you to mull over. If our classification systems are constantly being updated, as the chapter notes, why do you think it's so much harder to apply the concept of species to a microbe than it is to a mammal?
That very difficulty tells you something profound about our system.
It was designed by us for the world we can see, the galloping horse. And now we're trying to make it fit the molecular traffic inside a cell. What does that struggle tell us about the sheer, boundless adaptability of life itself?
It tells us that even with 1.8 million species cataloged, our work has really only just begun.